morning, everyone. I really do appreciate the opportunity to speak here at this beautiful venue. I'm from Australia. I live in California. That's more water than I've seen in my lifetime. So you should value it because it's a very scarce commodity in the places that I'm accustomed to being at. So it's a pleasure to be at the Dairy Innovation Hub, and innovation is the lifeblood of scientists, but it's also sometimes the controversy. And so today, I know this is a mixed audience. I'm going to assume there's some absolute experts in genomics and biotechnology, all the way through to people that maybe have never seen a cow. And I'm going to go a little bit through the science and give a sciencey talk. And then I'm going to segue in the second half towards science communication around controversial topics. And I didn't know growing up that I was going to work in animal agriculture and biotechnology, the actual trifecta, or the holy double of controversial topics, which I didn't know was going to be controversial when I first started. So I'm going to give you a little bit of a background as to why I have such a strange accent. So it's not from California. I do try to mimic it, but I'm actually from the country of Australia. And what you will notice from this graph here, or the slide, is that in the year that I was born, in 1963, you can do the math and figure out how old I am, there were 159 million sheep in the country of Australia and around about 11 million humans. So that was around about 14 sheep for every single human on Earth that was in the country. And you can see where the sheep are. I'm actually from this state, which is why I'm talking about sheep at a dairy innovation hub. And I actually think this was perhaps my favourite graph of Australia I've ever seen in my life. No sheep, some sheep. And really, that's all that matters. And the reason sheep are important is because really it was sheep that brought Australia into economic prosperity. That's why we're a first world country, is because we rode to prosperity on the back of a sheep, both for the meat that it produced, and back in the day for the wool, which was so important before the advent of synthetic fibres, if you will. And where these sheep lived is very much dictated by what land was available for them. And I think an often misunderstood aspect of particularly pastoral agricultural production is this land here, this light green land, which is grasslands unsuitable for crops that have animals. And that's their kind of secret niche, is where ruminants play a really important role in ecosystems. And that sometimes I think gets missed out, but as a result of the fact that they were able to utilize all this land that's otherwise unavailable for human food production, we had all of these sheep. And back when I was a youngster, I know my dad used to get breakfast in bed, because you did back in the 60s, and we would have fried sheep's brains for breakfast, and that's what you can see there. And we used to eat the brains and the chops and the liver and the tongue and the cutlets and shepherd's pie. And then the piece de resistance was always the roast on a Sunday with vegetables. And then there was always a good-natured fight about who got the lamb shank, because that was the very best part of the lamb. And that all was going on, and not to mention the fact that we have the merinos with their beautiful wool, which brought a considerable amount of income to the country. And when I first finished my agricultural degree, I actually worked in the sheep industry, and that's a picture of me from a few years ago now, with my friends, the sheep, working in a research setting, trying to improve their productivity. And actually I was working on a chemical defleecing agent. We don't have very many people, so there's not a lot of shearers, so we'd like ideally for their wool to be manually removed just by rubbing your hand over them. And this worked great. We gave them a pill and they defleeced themselves. Unfortunately, they defleeced themselves out in the pasture, and so it's the first time we'd ever had snow in New South Wales. And so, yeah, worked a little bit too efficaciously, but always trying to bring new innovations to agriculture. And so if we look today, there's only 70 million sheep. So we went from 159 million to 70 million. And if we look more generally at what has happened over time, is the human population has gone from 11 million to 26 million. The sheep population has dropped from 159 to 79, but those 79 million are producing more lamb, the valuable product, about the same amount of mutton, and the sheep consumption has dropped considerably on a per capita basis. And greenhouse gas emissions of a kilogram of lamb has dropped considerably as a result of the sheep being more productive. And we're bringing in considerable income in terms of both wool and meat to the Australian economy. And so from a sustainability perspective, looking at all of the aspects of sustainability, I think you could argue that that's a kind of an amazing all-around sustainability win for ag science in Australia. So simply put, Australians now consume about a fifth of the amount of sheep meat they did in '63. The sheep population has decreased by more than half, and those that are remaining are more than twice as productive, reducing the overall environmental footprint of a kilogram of lamb. And as a bonus, you get the wool. That's a great story, and that was kind of what I was raised in. And then I moved to the US and have been working in animal genetics and breeding at the University of California for a number of years. And I think this kind of success story is actually mimicked in many industries where innovation and technology has been allowed to improve the genetics of our systems. And so in the interests of the fact that I'm at a dairy meeting, I'm going to switch species now and move over to dairy cattle and just have a look at a similar kind of story that has been enabled as a result of the adoption of innovations in breeding and feeding and management and healthcare. But what we see here is similar to the story with the sheep in Australia. Back in the 1940s or so, there was around about 26 million dairy cows in the United States. And what we saw was over time, and going from the 1940s to 2010, we have a decrease in the cattle population, shown in blue here, and an increase in the yield per cow. And a lot of that was enabled by this AI arrow there. And I know there's a lot of younger people in the audience, and I do want to just put in a claim for the fact that we had AI before artificial intelligence came along. And so you may not take that acronym. That was always artificial insemination, and you guys are going to have to get a different acronym, all right? So, and nothing's more complicated than reading some of these new papers that are about AI of AI, and it's just like, I'm so confused. But you can do AI with AI. But really, the ability to utilize the very best male genetics and distribute it widely was one of the things, increasing the intensity of selection on those males, was one of the things that enabled the yield per cow to increase. And at the time, and I wasn't around in the 1940s, it was apparently quite controversial because it says it in its name, it's artificial insemination. And so there was pushback from the public around research that had to do with sex. There was pushback from the bull sellers who went, hey, hang on, you can buy the semen in a straw? What does that do for my business model? I might want to create a little fear around that. Oh, they're going to be Franken calves, you don't want to use that, that's not natural. But what actually enabled the technology to take off was demonstrations, often done by extension, showing that these calves were normal, healthy calves, and in fact quite productive calves because they had elite sires. And slowly the technology took off to where I would argue it's not a particularly controversial technology today. It's not 100% accepted. I see if you go onto Peter's websites and things, you see all sorts of crazy suggestions, but it's a pretty widely adopted technology in our animal breeding programs. And so what we see as a result of that, relative to the year 1944, the milk production per cow is more than fourfold increased. And as a result of that, there has been a knock-on effect of reducing the number of animals to about a fifth of the population that we had in 1944, eating less feed, drinking less water, using less land. And at the end of the day, you just want a really simple metric to tell your friends. The glass of milk today has about 1/3 of the environmental footprint that a glass of milk did back in the 1940s. And that is genetics, and there might be some nutritionists in the audience, so I better give a little credit to the nutritionist. There's probably a vet out there too. So how all of that together, but actually is a really, I think, an important success story around how adoption of innovation has aligned with many people's values on wanting to reduce the environmental footprint. And it is true that as a result of there being less dairy cows, there's less dairy farmers. And so there are always trade-offs associated with that for sure. But if the vision is to try to minimize our environmental footprint, I think it's undeniable that improved innovation has the ability to produce the same amount of product with less animals, and that, at the end of the day, has that knock-on effect of being desirable from an environmental footprint perspective. So that was the adoption of AI. Again, not very controversial these days. Similarly, since the sequencing of the bovine genome back in 2009, and I love that expression, the bovine genome, it was actually one cow, and she's a great cow, but there's many, many bovines on Earth, and so there's actually not the bovine genome, but a bovine genome was sequenced, which has enabled us to develop the SNP chips that enable us to select animals at a younger age and increase the accuracy of selection of those young sires, such that now we can introduce young sires into our breeding programs without having to go through progeny testing. That was kind of the magic that was enabled by genomic selection. And what we see is, as a result of kind of genotyping all of our bulls and getting the genomic selection prediction equations working, we've, using those young sires, have basically doubled the rate of genetic gain. And so you see, similar to the way AI had an inflection point where it increased the rate of genetic gain, so genomic selection has enabled basically a doubling of the rate of genetic gain relative to our net merit index, which is the selection index of dairy cattle in the United States. And so this technology, also not particularly controversial. I haven't heard too many people saying we shouldn't be genotyping our animals, or that that's unnatural, or they want non-genomic selected milk labeled for their production. So again, a really important innovation, enabled an inflection point change, but hasn't been associated with very much controversy. And more generally, we can see similar trends in food production from plant-based sources, and so this is a corn yield over time. You can see there wasn't much going on in the 1800s, early 1900s, but you start to see these inflection points as a result of the adoption of novel breeding technologies, the double cross, the single cross, and then in the case of plants, around about 20 years ago, they had access to genetic engineering, or GMOs, or genetic modification, however you want to think about it, which was for traits like insect protection and Roundup weed protection, which predicted yields, and you see another inflection point there. We haven't been able to use that technology in animal agriculture, and I'll get to that towards the end of the talk, and that is due to the public controversy around that particular breeding method and how that has really precluded the use of that technology in our systems, with the single exception of a fast-growing genetically engineered salmon that's been approved for food consumption in the United States. And so here we are in 2023 with a new technology that's come along, and something that my lab focuses on. And I'm going to talk for a period here about the science of genome editing, and then we'll talk a little bit around, are we going to be able to use it in animal breeding or animal agriculture more generally, or is it going to get stuck in the same problem that we had with genetic engineering? And so I'm going to talk about genome editing, which for those of you that are maybe not familiar with it, involves the use of a site-directed nuclease. CRISPR-Cas9 is the sexy one that most people talk about. There are other ones, Talens and zinc fingers, but everyone's kind of forgotten about them. And basically, they're just a fancy pair of scissors that introduce a double-strand break at a targeted location in the genome. And depending how that break repairs, you can either inactivate or knock out a gene, or you can provide a template and get an introgression of this blue DNA here. And so I can explain that to you, but most people get their information from YouTube. And so I am going to show you a video that tries to put that in context to understand what it is I'm talking about. So here we go.
For centuries, animal breeders have selected individuals displaying desirable qualities and bred them to produce particular goals. Breeders are actually selecting unnaturally occurring DNA sequence variations that result in differences between individuals, like the color of a cow's coat. Using conventional breeding programs, achieving desired changes can take decades, especially in large livestock species like cattle, which have long generation intervals. Innovations in biotechnology are poised to help breeders improve the rate of genetic gain. Genomes are made up of DNA, containing the collection of genes, unique instructions detailing every characteristic of an individual. What they look like, the amount of milk produced, or resistance to disease. Genome editing offers an opportunity to edit these instructions precisely. One method to edit genomes is the CRISPR-Cas9 system. This system has two components: the Cas9 protein, which cuts DNA, and a guide molecule that directs Cas9 where to cut. Bound together, they form a complex that introduces a precise cut in the DNA. The break is then mended by the cell's natural DNA repair machinery. This can be used to inactivate undesired genes, like those that cause disease. Alternatively, the repair machinery can introduce useful DNA sequences which result in desirable traits, like heat tolerance. Genome editing complements traditional breeding methods. It has the potential to introduce useful traits like disease resistance and adaptability into genetic improvement programs, addressing some of the problems facing global agriculture.
Now, I was a little bit nervous when I gave this talk, because I figured there would be some quantitative geneticists in the room, and they're quite scary to molecular geneticists. So I am actually contractually obligated to put up the Breeders' equation in any presentation I ever give. There's the Breeders' equation, quantitative guys. And I actually put this up here quite deliberately because I think genome editing is a useful tool to introgress simple traits. It will not replace quantitative breeders, it will not replace genomic selection, and actually it integrates with all of the tools we use in our breeding programs. And without genomics and assisted reproductive or advanced reproductive technologies, you can't do genome editing. And so I think it's always important to position it because basically genomic selection, which I described with the selection of using the SNPs markers and training population, enables you to select more accurately young animals and decrease the generation interval. That's kind of its magic thing, but basically you can then use that to select superior animals to multiply via advanced reproductive technologies, things like artificial insemination and embryo pickup and transfer. That also increases intensity of selection and decreases generation interval, but that merges with gene editing, which really requires a lot of these advanced reproductive technologies to come into existence. And so its power is that it potentially could introduce alterations that affect quantitative traits. They tend to be more polygenic and a little bit harder, but what its real magic is introducing useful qualitative or simple Mendelian genetic traits like horns, no horns, into animals that have elite genomics. And so you can introduce a trait that into your very elite germplasm that you want without having to do crossing with an inferior animal to bring it in. And that really is its power. And I always want to put it over, if you don't have a structured breeding program with a clear breeding objective like dollar net merit or whatever it might be, then the whole thing doesn't make any sense. And so this really sits within the existence of our current breeding programs. So, okay, with that background then, well, what might we knock out? Well, I just said that we're going to be working within our breeding objectives of our current breeding programs. Well, what do our current breeding programs select for? Well, they want a perfect adaptable dairy animal. They want one that not only produces milk, actually more ideally you'd like just protein and fat. So if you just plop out a block of cheese, that'd be awesome. Why even bother with the liquid, right? That would be cool. And in fact, I think the selection on liquid volume is almost zero in our current index. It's fat and cheese and reproduction and health and longevity and structure and all of the things that make you an optimal cow. And that, as a result, if you look at what we're trying to do here, we're trying to improve product quality, we're trying to reduce disease. Our breeding objectives haven't changed just because a new tool came along. It just gives us a new opportunity to potentially introduce useful traits. And I'm going to go through a couple of examples from cattle specifically. There are editing being done in other species, but really let's focus on cows because they're the coolest. And so what might we knock out? Well, here's an example from New Zealand. you'll notice this flag is subtly different from the Australian flag. It has red stars and ours are white. So basically, this is New Zealand knocking out the beta-lactoglobulin gene, which is a major milk allergen. So going down, that protein's not produced anymore. So that would be a product quality trait. Here's an example that's in the news at the moment, knocking out the prolactin receptor to mimic a naturally occurring mutation that results in a phenotype called slick. And slick is this very sheeny hair coat relative to this regular shaggy looking animal. And basically it keeps the body's core temperature down about 1 degree Celsius. And so it's a thermoadaptive trait. It'll help animals cope with hot weather. And that's work I know that's being done in like Florida and UC Davis with naturally occurring slick in dairy cattle. Well, what if you could introduce the slick trait into very elite dairy cattle genetics rather than having to breed it in from the Senepol, which is where it originated in the naturally occurring population. So that's a couple of examples of things we might knock out. What might we knock in? Well, again, our breeding objectives didn't change, so it's going to be the same traits that are important for our conventional breeding are going to be important. So let's have a look at disease susceptibility, unwanted development, like not growing horns, thermotolerance, improved food quality, and then I'll touch on one which is a sex ratio skew, which is one of the projects that my lab's been working on. So this, again, is an example from New Zealand, where they basically introduced a targeted alteration of this particular gene that makes dairy cattle black and white and made the brown instead. So that's a mutation that's mimicking a naturally occurring mutation in that color of cattle in other breeds. And again, this was done as a result or with the idea of brown not absorbing heat as much as black does. A lot of work going on in China. Sometimes hard to know everything that's going on there, but the stuff they've published has included attempts to produce tuberculosis-resistant cattle. Of course, that's a globally problematic zoonotic disease. And then just last week, this one was published, and this is work, again, with Recombinetics in collaboration with the Meat Animal Research Center. And I'll draw your attention to this animal up here. And I've actually discovered a phenotype that most people might be unaware of that enables you to tell whether an animal's been genome edited or not. And it appears to be they're always running with their tail straight up in the air. And I will support my statement with a couple more slides, but I can tell that that's genome edited just by looking at it. And basically what this did is a really interesting approach where they looked at a sequence where BVD, so bovine viral diarrhea, which is one of the viruses associated with bovine respiratory disease, so a big problem in cattle industry globally. And they said in this case, this CD46 protein, they hypothesized that if they did a targeted swap out of a piece of DNA in the middle of this gene, and altered basically the coding region. So, DNA makes RNA makes protein, and the protein is coded by the 3 base pairs. They basically replace the naturally occurring sequence of this gene with basically 18 base pairs that resulted in 6 amino acid changes in that protein, with the hypothesis that that would change basically the site where the virus gained entry to the cow and result in an animal that was no longer susceptible to that particular virus. So it's kind of using AI to some extent, or at least intelligent design, it's probably a bad choice of words too, design using understanding of how this protein works to then basically perform this work. And they used a gear, which is why that animal looks like it does. That's not because of genome editing, that's just what a gear looks like. And so they did somatic cell editing and then somatic cell nuclear transfer cloning a la Dolly, and then basically took that edited cell and produced this calf. And they've done some subsequent work, and they produced some controls and found out that these animals actually were resistant to BVD. So that's an interesting, it's one animal. I will note all of this work. There was one live calf. This is not quite ready to go to scale, and that's one of the challenges in terms of thinking about how would you introduce this breeding method into our breeding programs if we wanted to. So that's an interesting example. This is one you're probably familiar with. Can you tell if that's gene edited? What do you think? Gene edited? Yep, you've got it. Yep. So this again is work that was done by Exalogen, where basically they took it's a dairy cross animal. So yeah, that was from a dairy cross program at the University of Minnesota, and they replaced the the recessive growing horns allele of the pole gene. Basically, you replace that red box there, the 10 base pairs, with a duplication of this 212 base pair sequence, you get a dominant pole gene. In other words, the cow won't grow horns. And this particular animal got 2 copies of that, so he's homozygous for a dominant gene. And of course, if you cross a homozygous for a dominant gene, then you're going to get animals that don't grow horns. And so that is another targeted knock-in of this particular little 212 base pair duplication to introduce an allele that already exists in our beef cattle population, so Angus, for example, but you could bring it into the most elite genetics that exists in the dairy population and have that pole gene very rapidly dispersed through AI, which is pretty common, 70-80% of our dairy animals. So that's an example of where if you could edit an elite animal, you could actually introduce that trait quite rapidly into the dairy cattle population and eliminate the need for dehorning, which I probably don't need to tell this group is done to protect both human workers and the animals, and it typically involves either disbudding, and it's painful and unpleasant for the animal and the farmer, and it would be ideal if they were genetically not to grow horns. So this is work that was done in my lab, and yep, gene edited, yep, got it, see, I told you. So this is an idea of taking an existing gene, so the SRY gene on the Y chromosome, sex-determining region on the Y chromosome, is what takes the embryo down the male development pathway. So normally inherited on your Y chromosome. We copied it and inserted it onto chromosome 17, so that's an autosome, and produced this animal who was heterozygous for having the SRY gene, normally on the Y chromosome, on chromosome 17. And so this bull will then produce either Y-bearing sperm or X-bearing sperm, and then it'll 50% of the time get the SRY gene on chromosome 17 and 50% of the time won't. So in this case, if you've got a Y chromosome, you're going to be a male, so all of these male offspring are just going to be fertile normal males because they inherited the Y chromosome. If you get the X chromosome from this guy and the non-edited chromosome 17, you'll produce a normal female who's going to be fertile. But then this one is the interesting one. So you're going to be an XX individual, you've got an X from dad, but you also got SRY on the chromosome 17. And hypothetically, that will take it down the male pathway, even though it's an XX animal. It will be infertile because there's other genes on the Y chromosome required for fertility. And why would you want to do this? Well, in some industries, males are the preferred sex in terms of market animals for beef. So you ideally have animals that were all male going to the market. And of course, in the dairy industry, if you had your choice, you would like a bull that produced all females. And the laying industry also would prefer all females. And in fact, that would eliminate rather a problematic ethical concern of what happens to male chicks when they're hatched. They're basically killed on the day of hatch. So there's a lot of interest in how might you use this technology to produce single-sex offspring for the different industries where one sex is the preferred, or perhaps the only one that produces milk or eggs in the case of those types of industries. And so if you're interested and you want to watch a really detailed look at how we made this animal, there's a 30-minute YouTube video that we made that went through some of the stuff that didn't work too. I think sometimes we gloss over how hard some of this stuff is to get accomplished. But that was Cosmo, and he was born in the middle of the pandemic, which was fun because he was a bit of a big calf, and all of the drugs that you need for cesarean sections were being used to intubate people with COVID, and so we were a little bit nervous that we were going to have to do a C-section and we didn't have the right drugs, but anyway, he came out with a bit of a pull and he was fine. So I guess in summary, I would just like to say that, I think genome editing is kind of a cherry on top of the breeding sundae. So it does not replace all of the existing infrastructure we have for our breeding programs. It's really a cherry that enables you to introduce useful alleles without linkage drag. And that really is where its value is. And so you can have a sundae without a cherry, but it'd be nice if we could have the cherry. And so all in all, I'd say, it's a win for science. And if this was a scientific audience, it is, I'd be done. I'm done with my talk. Okay, any questions? But let's have a look at how this is going to play out. So this is the non-GMO project. Little butterfly, recognize that little butterfly? That's guaranteeing that your food with that little butterfly on it doesn't contain any GMOs. I don't really know what a GMO is, a genetically modified organism. I think what they're referring to is genetically engineered. But what they've done here is very sneakily said, oh, actually genome editing, even if there's no transgene or transgenic DNA introduced, we're going to call that GMO too. And so we're going to actually stigmatize gene editing products as being GMO too, because then we can sell more of our products with the non-GMO label. And so they're committed to preventing these new GMOs, that is genome edited products, from entering the non-GMO supply chain. And why does that ring alarm bells with me? Because I've dealt with this for my entire career, is the misinformation, fear-based campaign around scaring people around GMOs, that if you eat them, and you'll notice that the messaging is kind of goes to the heart of a parent's concern. So in the United States, they really focus on fertility and increases in allergens. And it depends which country you're in, which is the dominant narrative around what GMOs will do to you. So in Africa, for example, the real dominant narrative is it's going to affect male fertility. It's going to render you infertile. And that plays really well in Africa. And so they're quite strategic about how they try to cast this. And of course, there's been, I would argue, a targeted misinformation campaign to create the perception that these products are somehow unsafe or dangerous. And if you look on the internet, it doesn't take long to find anything along these lines that suggests that there isn't any consensus on the safety of these products. But as my good friend Abraham Lincoln says, don't believe everything you read on the internet just because there's a picture with a quote next to it, right? And when I see this type of thing, as a scientist, it's just in my DNA to go, but the global consensus of every scientific society in the entire world is that these products are safe. So how on earth did we get to a situation where we basically have the majority of the US population, so 63%, believing that these products are unsafe? How did the majority come to that wrong decision relative to what the science is? And it's actually the gap between where scientists are on the safety of GMOs relative to the general public, shown in red. That gap is greater than any politicized scientific topic, including climate change and childhood vaccines. This was done before COVID. I wouldn't mind doing it again. I have a feeling the vaccine one might have shifted a bit as a result of what happened during COVID. But why did that happen? And I'm going to quote a fella here who might have some familiarity to this particular audience, who says, some of the most polarizing topics in American politics are scientific ones, and even the existence of phenomena like global climate change and evolution are widely accepted in the scientific community, but questioned by large proportions of the US public. And as scientists, it's like, if only they knew, if only they had the information. It's that kind of deficit model. And unfortunately, interesting, don't know what that was, what we find is that, why is that doing that? Oh, I see what, I'm pushing the wrong button, that's what the problem is. Scientists have long believed it's just this information deficit, right? If only they knew, then they'd be fine. But actually, it's not just the science literacy, it's really the framing and the trust and emotional and social identity that affects whether people will listen to your message. And that's a real problem, because that means that the burden of scientific communication isn't the fact that the public don't know, it's that I'm doing a bad job presenting the information. And it shifts the burden to us and our institutions, our land-grant institutions, which is really disheartening, because it's much easier to believe it's just an information deficit than to say, I need to change my communication style if I actually want to have an impact in this particular field. And I think this framing is what we see with some of these controversial topics, is it's a very black and white framing. You're either with us or you're against us. So you either believe that GMOs are dangerous or you don't believe they're dangerous. And where is that dichotomous framing coming from? And I'm going to talk a little bit about my journey to the Dr. Oz show, which sometimes is held up as my, pivotal scientific achievement. And it's like, I write all these goddamn peer-reviewed papers, and I sit on panels, and I do all my community service, and that is what I'm known for. But anyway, I did go on the Dr. Oz show to try to really, it was as a result of the fact that we were having a proposition to label genetically modified foods in California. This was 3 weeks before the proposition, and it was just after the Seralini rat study had come out with these great big tumors on these Sprague-Dawley rats at 2 years of age that was suggested to be caused by the consumption of GMOs and the addition, strangely, of Roundup or glyphosate in their water, because I'm not sure why you feed rats that in the water, but these images were horrific. They were huge tumors in a rat strain that is well known to get tumors by 2 years of age, irrespective of what they ate or drank. It was the most cynical scientific experiment I think I've ever seen, because you knew what they would get, irrespective of what the treatment was. And that really went global. That one study resulted in entire countries in Africa banning all GMO imports. It basically created riots in France at the shipping docks when the products are coming in. So we went on there, myself and a colleague, and I'm basically going to show you a little clip now that is from a new documentary movie come out that's called Science Friction. And basically, it's an interesting documentary talking about, should scientists engage with the media? Should we allow our credibility to be used to push narratives that are incorrect? So does showing up at a debate with a flat earther give the flat earther credibility if you engage in that? Are you actually giving them credibility? And I'm going to play this clip that was shot after we had been on the Dr. Oz show to kind of talk about this black and white, with us or against us framing, which is the antithesis of scientific discourse. There is no black and white, especially in agriculture. Right? There's no one production system that's perfect. It's going to be dependent on so many variables. So I'm going to give you guys a rest and let the movie take it over.
I'm doing this show today to arm you with the information you need to make the best decisions about GMOs for your family. So here to help me make sense of it all are Dr. Pamela Ronald. She's a leading geneticist in the field of GMO plants. She's a professor of plant pathology at the University of California, Davis, and the author of Tomorrow's Table, Organic Farming, Genetics, and the Future of Food.
She's a colleague, and she had a pretty bad experience on Dr. Oz. She, again, was paired with Jeffrey Smith, who was in our interview also, and another individual from an activist organization.
Also with me is Jeffrey Smith, the executive director of the Institute for Responsible Technology and the author of Seeds of Deception.
Basically, the 3 of them were on stage simultaneously.
Dr. Ronald, talk to me about GMO foods and what you see as their future.
Okay, so at first it's helpful to know what we eat every day. So everything we eat has been genetically altered by some sort of methods. Yeah. Genetic engineering is another process, which is a very precise process. And indeed, one of the interesting aspects is you can take a gene from a different species and insert it. So if we can create a different type of agriculture, a more sustainable agriculture, by integrating our best farming practices and the best science, then we have made tremendous benefits to our children and our grandchildren.
The way that the interview went down, she really had very little opportunity to actually state her points or actually rebuke incorrect information that was given specifically by Jeffrey Smith.
Well, I work with doctors all over the world. I published a book called Genetic Roulette, the documented health risks of genetically engineered foods. We found 65. We found GMOs were linked to thousands of sick, sterile, and dead livestock, thousands of toxic and allergic reactions in humans, and damage to virtually every organ and every system studied.
Jeffrey Smith appears very honest, and he's a very good salesman, but he has no scientific expertise, and he's got some very bizarre beliefs. He believes in yogic flying, which is something like bouncing along on your ass and considering that you might take off from gravity or something along those lines. I mean, these are not alternative scientific opinions, they're alternative in a very, very dramatic way from where the evidence is.
The points that are raised by Jeffrey Smith have been widely refuted. I urge the audience to go to academicsreview.org, biofortified.org, ucbiotech.org. These are all university-based information sources.
So here's the problem. You know, I see this frequently in lots of different areas. The population that believes that GMO foods are safe will believe they're safe. The population that believes they're not safe is not going to believe they're safe. And it doesn't matter if you go and read a website. So convince me, I'm just sitting here, I'm asking, everyone watching this program sitting at home asking that question. They're saying either you're full of it or Jeffrey's full of it.
So we were a little nervous to go on Dr. Oz because she had had such a bad experience in terms of allowing the facts to come out.
In fact, that's part of the reason that we did eventually agree to go on, because it was to vindicate Pam's appearance there. It was so dreadful. And it was right after the Seralini study had come out as well.
There was a recent study that came out looking at rats who were given genetically modified food.
The Seralini's group fed 2 years of genetically modified corn to Sprague Dawley rats, which are the type of rat that every science experiment uses to study cancer risk.
The methods that they used, in addition to the fact that they did, use a rat breed that within 2 years, 80 to 90%, especially of the females, would have these gross tumors, irrespective of what you did.
And these are tumors coming out of these animals?
Yeah, most of the female rats got breast cancer. Most of the male rats got intra-abdominal cancer. There were problems with the kidneys, the liver, and the pituitary.
Now, if you're expecting that this is because of GMOs, you would expect a greater response the more you give. In fact, the animals that had the highest quote-unquote dose had the fewest tumors. And in fact, the animals that came out best were the ones that were given pure glyphosate in their drinking water. So go glyphosate, yay! But it was his show, it was his platform, it was his editing room. And I think no matter what we did, he was going to make sure it came out the way he wanted. And we were purely there as props to feed the position he wanted to have presented.
I think he went on before Congress and says,
I actually do personally believe in the items that I talk about in the show. I passionately study them. I recognize that oftentimes they don't have the scientific muster to present as fact.
oh my gosh, that's exactly what you have to have before you promote something to people. Otherwise, it's just snake oil. And he knows that. That's what I find perplexing as a scientist. It's like you've got nothing but your scientific integrity as a scientist. And
he sold his integrity. I mean, he may be a fine thoracic surgeon, but clearly he has absolutely no desire to be an honest broker when it comes to science.
We're not accustomed to dealing with people just telling bald-faced lies. We are trained to have scientific discourse with colleagues we disagree with, and we've all been in faculty meetings, we know there will be colleagues we disagree with, but we're not anticipating that our colleagues are just saying incorrect information. And I think it leaves us perplexed as to how to engage with someone who's just telling a bald-faced lie. And I'm not sure what these scientists are doing. This was stock footage when I Googled scientists, and I just said, is there a problem with the chicken? What are they looking at? This is the worst stock photo of my job. But I do think it's been very difficult for us to know how to handle misinformation, whether it's COVID vaccines or whether it's GMO misinformation. And typically, as we saw Pam diligently trying to get through on the Dr. Oz show, she was explaining the science behind GMOs and trying to appeal to basically the logos part of the discussion, so logic, reason, and proof. And that's typically, and pretty much today, the scientific background, the scientific presentation I've given you. little bit of background about genome editing, here's how we did it, here's the results, and here's some animals. That's how most scientists generally tend to structure their discussions and presentations. But there's actually two other really important bits here, which is ethos, credibility and trust. And I assume that you think, hopefully, that I am a professor at UC Davis, probably technically competent, not quite as competent as the ones at Madison, but relatively okay. And so I don't really need to establish that with this audience. The minute I walked up and was introduced as a professor, you had certain expectations about, like, I'm probably not going to be a yogic flyer that goes bouncing along on my ass. But then the one that we often miss in scientific presentations, and in fact is somewhat frowned upon if I suddenly start doing something that appeals to your emotion, is the thing that is actually really compelling from a science communication perspective, especially when talking to audiences that are not academic, because most audiences don't make decisions based on facts and reason, and that's really frustrating to scientists because we typically do. And so basically when you're giving a story to the public, you invert the typical way that we would present things and start off with shared values and the bottom line. So the so what, and then maybe they're interested in whether or not you used a homologically directed repair template in your repair pathway to produce your bull that doesn't have horns. Really, they want to know, why would you want a bull without horns? And it doesn't really matter what technique you use to produce that. But I think what has happened, especially with social media, is that the very powerful narrative as it relates to persuading people is really to go strong on that pathos or emotions and values. And many of the techniques you hear, which work well, storytelling and anecdotal information, get kind of, I guess, hijacked, for want of a better expression, to create fear, and vivid language to create a narrative that suggests that someone, our common enemy, Monsanto, big cigarettes, academia, big meat, big dairy. Is there big dairy? Big dairy, I'm sure there's big dairy. Big cheese, I guess, I don't know, for want of a better expression. And what you'll see is people like Jeffrey Smith, who was in the Dr. Oz show twice, both in my session and the session before with Pam, who has absolutely no academic criteria whatsoever. I think his training is as a swing dancer, is on the #1 watched national television program as an expert. And, well, look, he's the executive director. Well, that sounds pretty impressive. At the Institute for Responsible Technology. Well, who wouldn't want to work there, right? As distinct from the Institute of Irresponsible Technology down the road here, right? And he's got 2 self-published books, and he documents the 65, very precise number. So he kind of does a little bit of logos and ethos, but really the main thing is the pathos. And so how do we go about countering this? And I think that that's a really hard skill set for academics who've been trained to show no emotion whatsoever, to actually switch into a more human mode of interaction and try to figure out, what are my shared values with this community? And so I think that we actually, if we want to use these technologies, we need to get out there and be active in communicating it to the general public and taking some risks. And I will just say that if you're interested, this movie Food Evolution was produced a few years ago now. It's freely available on Hulu. I used to have to explain what Hulu was, but after the pandemic, I don't need to explain Hulu anymore. But basically what this was a collaboration between academics and narrative storytellers, like documentary filmmakers, who know how to connect to normal human audiences and basically have the skill set that's required for effective translation of these complicated topics to the general public. And I think this particular movie, which was narrated by Neil deGrasse Tyson, really was a useful public engagement tool to reach a bigger audience than we typically do, in a medium that a lot of people get their information from, which is movies and videos. And so how do we engage more in these types of public discourses? And I have certainly been active in this area, and sometimes with success. So we had the Washington Post do a story actually on some of the genome editing we were doing to make Cosmo, the bull that had the extra SRY gene. And they did actually a really nice story. It was balanced. It was like they're coming, and then it gave a nice presentation of why we were doing that and will we eat them. So it discussed those controversies. And they came along and took pictures of us. We're actually doing an ET here. You can't see the other end, but that's what's happening here with the vet. Very representative of what was going on. That then got picked up by an Australian newspaper, so the Australian Financial Review, and it had the same verbiage here, but they introduced pictures that they thought would better illustrate this particular story, which was about genome editing in cattle. And so the first image is fine, we've got some cows there, that's good. So cows are being altered using CRISPR, a technology that allows scientists to make targeted cuts to the genome and change or insert new genes, that's all good. But what's going on down there? What the hell's that? I literally have no idea what that was. Or why the heck that was inserted in the middle of that story. So you have some losses, but I will say, and I don't have time to show you this piece, we had Nova out to do a story on Cosmos, so again, the SLY bull that was born. And the reporter that came out to do it was a medical doctor from San Francisco who was of Indian descent and didn't eat beef. I thought, well, this is going to be fun. He did the best segment you've ever seen in terms of integrating humor, explaining it in a very captivating way, and actually presented it with the concerns and the discussion around editing in animals and humans. That was just, it made my heart sing. And this was obviously a video. This is, if you don't know, is a documentary series that PBS Nova has. And you can see him here, he's pretending to sneak up on Cosmo. And when he was talking to me before he came out, I said, make sure you wear boots or, something, because there'll be manure and stuff around. And he gets out of his car in just the most preppy pair of white sneakers you've ever seen in your life. And he knew exactly what was going to happen, and he played it up. So eventually he ended up getting shat on, and he got shit all over his white shoes. And it was a really funny thing that the general audience of watching Nova would totally relate to. And so it brings in the kind of the humor, which is so important for science communication, but did such an amazing job telling the story in a way that I wouldn't have necessarily done, but I think these partnerships with people that are able to tell stories in a compelling way is a really important thing, so that we do have some of this information coming out in the general public, so it's not catching people by surprise when they first hear that we're actually doing genome editing in cattle. So with that, I thank you for inviting me. I know it's been a bit of a wild ride from science all the way over to science communication, but if we don't have the ability to use these technologies, then why bother actually with innovations if actually we can't ever get them adopted into our animal agriculture. So thank you, and I'm happy to take questions.
That was amazing. For time's sake, we'll have time for one question. I know that's not fair, but just time for one question. So who is the lucky person? Back there, sir. Just 1 second. She'll bring a mic to you.
Australia has the best sheep meat. How does it come to that?
Well, I hesitate to say it, but first of all, our definition of a lamb is quite different to the American definition of a lamb, so they're much younger animals, and they don't get ever finished on corn. They're coming either straight off their mum or off grass, and I like that flavour, some people might not, but that's, I think, part of the reason. Also, they've got a pretty heavy selection objective now for meat quality, and so it has been a result of intentional breeding for meat quality as well.
All right, well, I think we all have our assignments for Hulu watching, some YouTube deep dives. I know I'm going to be checking those out. So let's thank Alison one more time for an incredible talk. We're going to be transitioning now. We have a couple of breakout sessions. They are up on the 3rd floor. There's 2 of them. And also there are posters that as you walk down the hall, those are available at all times. So in the next several minutes, if you could kind of make your way to the third floor. We will have those two events. And then there will be, this afternoon, we'll be having some other exciting talks. There's a flash talk competition. So please grab a snack, check out the posters if you'd like, but if you could make your way by 10 o'clock up to the third floor to those breakout sessions, greatly appreciated. Thanks.
I didn't even touch on that.
So have animals never really been the same? Yeah, I guess the USDA does regulation based on plant and human. Yeah, right? So I guess animals maybe falls like under like. Yeah, so maybe it's a possibility that it will get.
Well, there's an executive order to make that happen, or
Excellent as usual. You've inspired me. I've had to push back. Yeah.
Everything worked well.
Glad to hear it.
Once we got through the initial snubbles.
Right.